This presentation begins by examining coffee's seemingly contradictory relationship with genomic integrity. On one hand, it contains compounds with theoretical genotoxic potential. On the other, it delivers a diverse array of bioactive constituents that may offer protective effects. To understand this duality, we rely on the established OECD genotoxicity testing framework, guidelines 471 through 490, which evaluate mutagenicity, clastinogenicity, and DNA damage across multiple experimental systems. Coffee is far more than a source of caffeine. Roasting generates over 1,000 bioactive compounds through Maillard reactions and pyrolysis, including methylxanthines, diterpenes like caffeistol and convoyol, chlorogenic acids, melanoidins, and numerous heterocyclic molecules. This chemical complexity challenges reductionist toxicology because testing isolated compounds rarely captures the integrated biological response elicited by the full beverage matrix. Acrylamide forms during roasting when asparagine reacts with reducing sugars at temperatures above 120 degrees Celsius. Although acrylamide is considered a probable human carcinogen and neurotoxin based on rodent data, coffee contributes only a small fraction of dietary exposure, around 0.45 micrograms per cup, compared with fried potato products, which can contain 39 to 82 micrograms per serving. Darker roasts contain less acrylamide due to thermal degradation, demonstrating how processing conditions shape contaminant levels. In vitro studies show that extremely high, non-physiological caffeine concentrations can inhibit RAD51-mediated homologous recombination and trigger cell cycle arrest. However, these effects occur at levels far exceeding those achievable through normal consumption. This distinction highlights a core toxicological principle. Cellular responses observed under artificial laboratory conditions must be interpreted in the context of realistic human exposure. Coffee's protective effects can be understood through the lens of hormesis. Rather than acting solely as antioxidants, phenolic acids and diterpenes activate the NRF2 signaling pathway, which upregulates phase 2 detoxification enzymes such as glutathione S-transferases and NADPH-quinone reductase. This adaptive enhancement of cellular defenses aligns with OECD guidance, acknowledging that mild, transient stressors can induce beneficial biological responses not captured by traditional hazard-focused assessments. Human intervention studies using the COMET assay, validated under OECD Test Guideline 489, show that coffee consumption reduces spontaneous DNA strand breaks by 23 to 35%. This decrease in baseline DNA damage supports a genoprotective effect under typical consumption patterns. Because the assay is sensitive to oxidative lesions, it is particularly informative for evaluating beverages rich in redox active compounds, though it should be interpreted alongside complementary genotoxicity endpoints. The diterpenes caffeistol and convoyol illustrate coffee's dual nature. They strongly induce glutathione S-transferases, suppress carcinogen activation, and exhibit chemopreventive properties across multiple tissues. At the same time, they can raise serum LDL cholesterol by altering bile acid metabolism. Brewing method determines exposure. Unfiltered coffee contains 3 to 6 milligrams per cup, whereas paper filtration removes most diterpenes. Balancing risks and benefits depends on individual cardiovascular profiles and consumption habits. Caffeine metabolism varies widely across individuals due to CYP1A2 polymorphisms. Fast metabolizers, those with the AA genotype, clear caffeine efficiently and may experience cardiovascular benefits, while carriers of the C allele metabolize caffeine more slowly and may face higher hypertension risk with heavy intake. This pharmacogenetic variability underscores the importance of accounting for population heterogeneity in regulatory safety assessments, consistent with ICH M3R2 guidance. Brewing technique significantly influences exposure to key coffee constituents. Paper filters remove diterpenes, supporting cardiovascular safety for individuals with elevated lipid concerns. In contrast, metal mesh and immersion methods allow diterpenes to pass through, potentially enhancing chemoprotective enzyme induction in those with normal lipid profiles. These methodological differences represent important exposure modifiers that should be considered in human studies. Roast level affects both contaminants and beneficial compounds. Dark roasts contain less acrylamide due to thermal degradation, while generating higher levels of NRF2 activating melanoidins and pyrolysis products. This inverse relationship between harmful and protective constituents illustrates how processing can simultaneously influence multiple exposure pathways, complicating simple hazard-based interpretations. Taken together, the evidence supports a net genoprotective effect at moderate consumption levels of roughly 450 to 600 milliliters per day, about 3 to 4 cups. Within this range, human biomonitoring studies consistently show reduced DNA damage markers, enhanced detoxification capacity, and negligible acrylamide exposure relative to toxicological thresholds. These findings align with the International Agency for Research on Cancer's conclusion that coffee is unclassifiable for carcinogenity following comprehensive review of epidemiological and mechanistic data.